So when the guanine becomes oxidized, it can eventually form a lesion known as the 8-oxoguanine, which is like one of the most common forms of the oxidative DNA damage. So if this 8-oxoguanine is left unrepaired, such lesions can lead to mutations and a genomic instability as such. So cells normally correct this damage through the base accession repair strategies. I mean, the BER enzyme pathways as such. Traditionally, the DNA repair has been viewed primarily through the lens of biochemistry. However, the recent experimental findings have raised an intriguing possibility like could quantum spin dynamics also influence these repair processes as such? So this is the main question that was the motivation behind our work as enough. Recently, we found a paper which directly has told that they have observed a long-living radical pair between the flavin and the guanine in the artificially constructed DNA oligomers. So, what makes this discovery remarkable is that this radical pair survives long enough to exhibit the measurable magnetic field effects even at the room temperature, which is quite unusual for the biological systems. So, our study investigates whether this flavin guanine radical pair could function as a spin sensitive intermediate or a byproduct in a DNA repair signaling. So specifically, we ask, we are questioning whether this magnetic fields and the chirality, the molecular chirality, can influence the outcome of this RPM reaction, and thereby that modulates the repair related signaling pathways. So, before discussing our model, I would like to introduce the brief content of the radical pair mechanism in our essence. So, the radical pair consists of two molecules, an unpaired electron. So, these electron spins are quantum mechanically correlated and can exist in either singlet or the triplet stages. So, in our flavin guanine system, the radical pair is formed at the triplet stage, where the guanine acts as a donor and the flavin acts as an acceptor. So once formed, the spins evolve under the influence of this internal hyperfine interactions with the nearby nuclei and the external magnetic fields as such. So as a system goes on, the interconversions can occur between the singlet and triplet spin configurations since the chemical reaction pathways often depend on spin states. So the final reaction products can be influenced by these electron spins and its dynamics. So it was studied that the singlet pathway leads to the charge recombination, which further leads to damage, and expected base accession repair strategy is sort of achieved in that way. And if we consider if the reaction is heading back to the triplet pathway, the radical escape happens, which doesn't trigger the enzyme repair strategy, but thus we can say that the DNA is set to heal itself via the quantum phenomenon. And an additional ingredient in our work is about the cis activity, the chirality induced spin selectivity, where actually we have learned that the electron transport is happening via the DNA medium. So the biological molecules such as DNA, this possess the chirality, the chiral structures in their stuff. So according to the cis effect, the chiral molecules can preferentially interact with the electrons of typically of one spin orientation over the other, and thus they will act as a kind of a spin filters. So this means that the whole chirality of the molecule may bias the whole RPM dynamics, and that can alter the reaction outcomes as such. So therefore in this work we combine the main elements, as in like the first is the long living flavin guanine radical pair which is observed experimentally, and the second point is the magnetic field effects on this spin evolution. And the third main point is to address about the chirality induced spin selectivities. So using this framework, we have investigated how these factors influence the balance of repair signaling versus the recombination pathways in the DNA damage responses. So with that said, that we have established the biological and physical background of the study, I will now want to hand over to my colleague Ashutosh, who will explain the methodology, the theoretical model, and the master equations behind and the formulation as such. Over to you, Ashutosh.
Thank you, Akshaya. So, good afternoon, everyone from India. So, we use the spin Hamiltonian, which consists of four interactions, notably the Zeeman, the hyperfine exchange, and dipolar interaction. The system evolves under this specific Hamiltonian, where symbols hold their usual meanings. As the system starts in triplet state, we take the summation of t naught, t plus, and t minus states as our initial state. And the master equation, which is like visible to you on the screen, it consists of, as Akshaya told, that only the singlet pathway can recombine. So the anti-commutator, which basically leaks the trace out of the system, which means that like in our initial density matrix, information is leaking out via the singlet channel, while information can also leak out into in the forward direction where both singlet and triplet participate. So that's why there's another term called KF times rho t. This solution of this master equation will evolve our initial density matrix in time, and that evolution can be thought of as a unitary operator acting on the initial state, and that unitary is defined by the effective Hamiltonian, which is visible to you. So this effective Hamiltonian is nothing but an analytical solution of our Lindblad master equation. The yield in any direction is given by the rate multiplied time integrated singlet expectation, uh, which we will get after roti. The singlet expectation is after evolving the system in time. When we get the density matrix at time t, we take the trace of rho into an operator, and this is statistically it will tell us about the expectation of singlet at any time. We feed this into the time integrated average and multiply it with the rates to get the basic probabilities of whether the initial system will choose the backward pathway for recombination, or it will choose the forward pathway for forming spin independent products. I'll hand over to Akshaya to explain you about the results. Thank you.
Yes, yes. So now moving to briefly about to our results, as you see, so our simulations have showed a clear trend. As the degree of the cis-induced spin filtering increases, the forward signaling yield decreases. So in other words, the stronger chirality tends to to suppress the signaling pathway as such. So this suggests that the enhanced spin selectivity promotes the recombination and the repair related pathways rather than the signaling pathways, which is prone to the furthermore BER strategies as such. So we therefore observed a negative correlation between the chirality strength and the repair signaling yield, is what we tend to say. We have also investigated the effect of changing the direction of the external magnetic field within the parameter space explored in the study. So the magnetic field dependence was relatively weak. Some variation was observed at lower chirality values, but this directional sensitivity gradually disappeared as the chirality increased. So overall, our results have suggested a potential connection between the DNA damage and the radical spin effects and the whole chiral quantum phenomenon. They also provide a framework for exploring this experimentally testable quantum signatures in the DNA repair processes as such. So uh, with that, I would like to talk about the future works and the possible applications that we would like to move forward from our present studies. Is that to extend this the model towards the disease relevant DNA repair regulation studies as such, and possibly also to develop a framework as a basis for the magnetic field based biosensing or the chirality sensitive based biosensing for DNA damages, and also to know about its repair states as such. So with this, I conclude our research work as in the poster. So I'm thankful to the QEPS and the ELT University for the opportunity, and this is it.